Hey Mustangs, in this video we're going to be reviewing DNA replication. So first thing, a little bit of background information. Taking a look at this image here, we're looking at the antiparallel structure of the DNA double helix. Here we don't actually see the DNA double helix itself, but we can see the two different strands. So there's a strand on this side here, here's a second strand here, and the antiparallel nature is key to really understanding DNA replication. So we have the 5' end here on this strand, the 3' end here, and when we say it's antiparallel, this side has the opposite, 5' end here, 3' end there. Remember, when we're talking about the ends, 5' end is the end that has the phosphate, 3' end is the end that does not have the phosphate, it has the OH group instead. Another key bit of background information is going to be the fact that anything that ends in ASE represents an enzyme. So there's going to be several different enzymes that are involved in DNA replication. So the question is, how do you get from a single cell with one copy of DNA instructions to a multicellular organism that has trillions of cells, all with the same DNA instructions? Now the key is DNA replication. So in DNA replication, it's making an exact copy of all the DNA inside of a cell to make sure that when the cell divides, each new cell has the exact same DNA instructions. So if you take a look at this cell here, this cell has a single copy of all your DNA instructions. If you remember, humans have 46 chromosomes, so you have all 46 chromosomes in here. When that cell divides, as we see happening here, the two new cells that form are going to have 46 chromosomes in them. Both of them will have the exact same DNA instructions. So as cells keep on dividing, every single time a cell divides, you make a copy of all the DNA instructions and both cells have the exact same instructions. Now if you remember, cells become different or differentiated or specialized when certain parts of the DNA instructions get turned on or off. So looking at these here, at this point, these are stem cells. These are cells that haven't been specialized yet. They haven't gone through cell differentiation to become different types of cells. All right. Now, there were 3 proposed models of DNA replication. The first is the one that is actually accepted today. So semi-conservative model. With the semi-conservative model, you wind up with 2 new DNA molecules at the end of DNA replication. Each molecule, you can see in blue, that's an original strand, and then the yellow represents a brand new strand that was formed based on the original template strand, the original blue strand. And then same thing here, you have an original strand and a brand new strand, and that's a semi-conservative model. The conservative model had the idea that you can use the original strands as a template to make new strands, and once they were used, then the old strands got back together and the new strands got together. So that was the conservative model. And then finally the dispersive model was kind of a mix here of old strands together, new strands all in one single molecule. So again, this is the one we accept today, the semi-conservative model. So if we take a look at a few different views of the semi-conservative model, here we have a DNA double helix. It separates, and these strands, these original strands, can be used as a template to know what to lay down next based on the base pairing rules. So we know we'd have to put a nucleotide here that would match a T, so it would be an A, then a G, then a T, then an A, and so on that side. And then same thing here, you'd use base pairing rules. A goes with T, G with C, and you lay them down just like you see here. So you can see the white is the original strand on both sides, and then the green would represent the new ones, and here's our semi-conservative model. 1 old strand, 1 new strand each. And one more time. So here we have the original DNA molecule. The old strands are used as templates. So original strand, and then you have the new strands that attach. So DNA nucleotides come and form the new strands. Now, the first step, the more complex than just simply laying down the nucleotides in DNA replication, the first step is unzipping the DNA. So there's 3 big things that you want to see here. So there's helicase enzymes that do the unzipping. Then there's single-stranded binding proteins and topoisomerase. These 3 things are all involved in unzipping the DNA. So the helicase enzyme goes through and breaks hydrogen bonds in order to separate the 2 original strands. When the bonds become separated, it forms a replication bubble with replication forks. The single-stranded binding protein's job is to make sure those strands don't come back together. Topoisomerase its job is to go further down the DNA molecule and relieve tension, because as the two strands unwind, it causes tension further down in the DNA molecule. So if we take a look here, we have, here's DNA, antiparallel here, 5' 3', 5' 3'. We have an origin of replication, so there's a point in the DNA where basically there's certain A's, G's, T's, or C's that are the initiation. That's the origin of replication, where DNA replication will start. At that point, this is a replication bubble. So as DNA helicase, starts unzipping the two strands, this is a replication bubble. This is a replication fork. This is a replication fork. Same thing here. Here's our replication bubble. That's a replication fork here. That's a replication fork here. Now, taking a look at this puts the things together that we've talked about here, adding a little bit more information. This will be with actually building the DNA, so we can kind of ignore the primase and the RNA primer for now. So we have helicase enzyme that's doing the unzipping, and it even looks like a zipper here when we look at this image. So helicase goes through and unzips the two strands. Topoisomerase relieves tension further down, and the single-stranded binding proteins seen here in gray are going to be used to make sure these two strands don't go back together. All right, now adding the nucleotides. It's actually a really simple process in one way, but difficult in another. So if we take a look at adding nucleotides, you're going to need RNA primase. The ACE tells us this is an enzyme. So RNA primase is going to create RNA primers. DNA polymerase 3 will be the enzyme that actually adds the nucleotides. And then this part right here, it's built in the 5' to 3' direction. So DNA can only be built in that direction. So if you have a nucleotide, like we see here in this simplified drawing, okay, really simplified drawing, this is the 5' end, and this is the 3' end. So a new nucleotide can only be added over here in order to build the DNA. You can't add going up the other direction. So you can only build DNA in the 5' to 3' direction, adding on down here with the nucleotides. So if we take a look at our image here, the RNA primase is going to add an RNA primer. So what you see right here in red is actually not DNA. So blue, this dark blue is the original DNA strand. This red represents the RNA primer that's been created. So RNA primase lays down the RNA primer. From there, the DNA polymerase 3 can be used to add nucleotides from that point on. So this pink molecule you see here is actually DNA polymerase 3 adding new nucleotides on from the 5' to 3' direction. Now, going back here, the RNA primer must be replaced by the end of DNA replication. So another molecule called DNA polymerase 1, so Pol1, oops, went past it there, let me go back for a second. So here we have DNA polymerase 3 that's adding the nucleotides on, but this needs to be replaced. These are RNA nucleotides, and you want DNA nucleotides in there. So that's where DNA polymerase 1 comes in. So DNA polymerase 1 will eventually replace these with DNA nucleotides instead of RNA nucleotides. All right, so taking a look at this, if we add the nucleotides on, you're going to see that the RNA polymerase 3 adds nucleotides. Here's the 5' end. There's a 3' end, so it's going to add it that way in the 5' to 3' direction. So it just goes through and keeps on adding on nucleotides. Now, quick review question. What type of reaction are we looking at here? Is building DNA anabolic or catabolic? And is it exergonic or endergonic? So because it's building, it's anabolic. And anabolic reactions require energy, so it's endergonic. All right, so where's the energy coming from? If it's an endergonic reaction, energy is being put in, where's the energy coming from? So we say DNA is made out of nucleotides, but they don't come in as nucleotides. They actually come in as nucleosides. And a nucleoside is simply this. Instead of one phosphate, there's actually 3 phosphates connected to it, like this. So instead of a nucleotide, we have a nucleoside. Now when these 2 phosphates break off, that provides the energy to add and build the DNA. So to add new nucleotides on and actually build the DNA. So if we take a look at our example here, here comes DNA polymerase 3 with the nucleoside. Notice that it has the 3 phosphates 2 phosphates are broken off, and that provides the energy to keep on building the molecule. And that just keeps on going all the way through. All right, now there's a problem. When you take a look at it, on one side, we can build continuously in the 5 to 3' direction. Okay, so on this side here, we see we can build 5 to 3' continuously. No problem, just keep on building it. But on the other side, if we tried to build in the same direction, so notice this one's going this direction. If we try to build this side in this direction, that doesn't work. So if we do that, we're actually going 3 to 5', and DNA can never be built that way. So here you try to put down your first nucleotide and then add the next nucleotide here. It's not going to work. So you try to do that, nothing happens. So it can't be built that direction. So we see that it has to be built in the 5 to 3' direction. So let's say we start here and we build in the 5 to 3' direction, then that works. Now, because one side can be built continuously, we call this the leading strand. The other side that has to be built in little fragments like this. So if this starts building here, you're also going to have to start building down here, and you build in a whole bunch of fragments. Those are called Okazaki fragments. Now, in order to connect those Okazaki fragments together, we use another enzyme called ligase. Ligase actually connects those Okazaki fragments together. So if we take a look here, leading versus lagging strands, they both have to be made in the 5 to 3' direction. The leading strand is made, as we see here, from the RNA primer, gets made in the 5 to 3' direction. And the lagging strand, if we take a look at this, here's our leading strand, 5 to 3' continuously. Here's our lagging strand having to be built the opposite direction in 5 to 3' direction. You have to use many RNA primers to make the Okazaki fragments. So here's an RNA primer, here's another one, there'd be another one here, and you just keep on adding these primers, and the RNA primase will build the primers, and then the DNA polymerase 3 is going to do its job just like it did here. So it's going to add nucleotides here. The DNA polymerase 3 will get all the way to the next primer. When it gets here, DNA polymerase 1 will replace these RNA nucleotides, and then ligase will connect the strands together. Okay, so again, RNA primase creates the primers, DNA polymerase 3 adds the nucleotides. When it gets here, RNA polymerase 1 replaces the RNA nucleotides, and then ligase connects the strands together, the Okazaki fragments together. So here's all the different things that we see here that we've learned so far. Right, so here's the overall steps for elongation of the lagging strand. So basically exactly what I said using the images. So RNA primase adds RNA primer, DNA polymerase 3 adds the nucleotides in the 5 to 3' direction. It detaches when it reaches the next primer. Polymerase 1 replaces the RNA primer with DNA nucleotides, and DNA ligase joins the Okazaki fragments. All right, now there is proofreading that goes on. So as the DNA replication occurs, it makes sure that it doesn't make mistakes. Mistakes mean that proteins will not be built correctly. So this has to be a very, very well thought out, well done process. So as it goes through, proofreading occurs, but if there are any mistakes, there's mismatch repair and also nucleotide excision, which could actually correct any mistakes that were made during replication. Now, like I said, mutations are results usually when DNA replication goes wrong. So it puts down the wrong A's, G's, T's, or C's, and it creates a different set of instructions for building a protein. If the protein gets built and it's functional and it can do something, then that can lead to a mutation that can either help the organism or hinder the organism. And then replicating telomeres, the ends of DNA. Every single time you make a copy of your DNA, you are basically causing the ends to become shorter and shorter and shorter, because the DNA polymerase 3 doesn't go all the way to the end of your DNA molecules. So every single time copies of the DNA is made, the ends, called telomeres, get shorter and shorter and shorter. Now there are enzymes that actually can elongate those ends, and if we can actually figure out how to do this, we could basically cause a cell to technically live forever. So cells can live a lot longer as long as their DNA doesn't get shorter and shorter over